glycogen storage disorders. So in glycogen storage disorders, the first step, we look at the cycle. Glucose to glucose 6 phosphate, it is converted to glucose 1 phosphate, and then the cycle begins. Looking at the cycle, glucose 1 phosphate, it is converted to the UDP glucose. This is converted to amylopectin. Now in these reactions, the enzymes involved is, here it is UDP glucose pyrophospholase. Next, what we see is glycogen synthase. Next, the amylopectin, with the help of branching enzymes. Next conversion we see is glycogen. Then it is going to limit dextrin. Next we see it is going to glucose 1 phosphate. Now the branching enzyme, the glycogen here goes to glucose. This is a process of the lysosomal degradation. Now looking at the diseases, first finishing the enzymes, this is by debranching enzyme. This is in case of glycogen to the limiting dextrin, it is glycogen phosphorylase. Now looking at the disease involved, in case of the conversion of glucose to glucose, glucose to glucose 6 phosphate, here the enzyme, the disease is von Gerke's disease. Next, looking at the other disease is Pompey's disease. This is where the glycogen is converted to glucose. This process of lysosomal degradation. So Pompey's disease is involved here. Disease. Next, looking at the McArdle disease. This McArdle disease is basically the conversion of glycogen to limit dextrin. So in between this is where the McArdle's disease is present. And the enzyme here was glycogen phosphorylase. Now next, looking at the limited dextrin, the step of limit dextrin to glucose 1 phosphate. So in this step, the enzyme involved is a debranching enzyme, and the disease is the Cory's disease.